to talk about the second half of our lecture on viruses. We're going to cover the second life cycle in bacteriophages, the lysogenic cycle. And we're going to cover animal viruses, mammalian viruses. And if we have time at the end, I'd like to take a look into plant viruses and prions and viroids. But I don't know whether or not we'll get into those, so we're going to kind of play that one by ear. Here we have viruses part 2, the lysogenic cycle. So last time we said we talked about the lytic cycle, and we said that all of your standard normal bacteriophages can do the lytic cycle. And some viruses can only do the lytic cycle, and they're usually referred to as virulent phages. There's a different type of phage, a different classification of phage, called a temperate phage. Temperate phages can do the lytic cycle, or they can do the lysogenic cycle. So, temperate means sort of moderate or controlled. Like, the temperate climates are not tropical, they're not Arctic. If you say somebody has lost his temper, what does that mean? That means that they've lost control. Whereas, like, the temperance movement back in the 20s was people who abstained from alcohol so that they could constantly be in control of themselves. So a temperate phage is one that has control. It has control over which cycle it's going to enter. It could enter the lytic cycle, or it could enter the lysogenic cycle. Who remembers the first stage, the name of the first stage of the lytic cycle? Attachment. How about someone from over here, the second stage? Penetration. The third stage, or third and fourth, kind of together. Not yet. All right, someone from back here. Do you remember? You're absent, right. Pardon? Synthesis and replication. Next comes assembly. And the last stage is release. The lysogenic cycle starts like the lytic cycle. In fact, it's sort of a subset of steps that can be tacked on to the lytic cycle. So the first step, again, because it's going to start out exactly like the lytic cycle, is going to be attachment. Phage attaches. Step 2 is, again, going to be penetration. So in this, so far, steps 1, steps 2, exactly the same. Step 3 is where it's going to start to diverge. Step 3 is integration. During integration, and we talked about this, we sort of hinted at this when we were talking about specialized transduction. What you saw here, watch this viral genome as it goes in. And you'll see the viral genome is going to sort of disappear as it kind of melds into the bacterial genome through the process of non-homologous recombination. It's sort of, there's a little snick and a little snick, it gets cut. And this part of the bacterial genome gets attached to the viral genome, and this part of the bacterial genome gets attached to the other half of the viral genome, and the viral genome inserts itself into the bacterial genome. This step is called integration, and once the viral genome has integrated, it's now referred to as a prophage. It's still a virus, but now it's also a part of the bacterial chromosome. It's a virus that's living inside of the bacterial chromosome. Now we said that lysogenic or temperate phages have control. They can decide to go lytic or lysogenic. What's going to influence this decision is the health of the cell. So if they get into a cell and they go, hey, you know, this cell is kind of a crappy cell, it's not dividing very quickly, I'm just going to, you know, replicate a bunch of times and then kill it, and I'm going to go look for a better cell. It's not a very good cell, I don't want to stay here for very long. Then it goes on the lytic pathway. But if it gets inside of the cell and it says, hey, this is a pretty nice place. This cell is all nice and healthy. It's dividing like crazy. It's a, you know, this is a nice place. This is a nice neighborhood. I'm going to settle down here for a while. So it makes its decision based on the health and reproductive speed of the cell. Cells which are growing fast and reproducing quickly, those are the sorts of cells that are going to get targeted for integration. So step 4 in the lysogenic cycle is reproduction. Now, in this, what's happening is the bacterial cell is reproducing, just the same way that bacterial cells always reproduce. It's just doing the normal bacterial reproduction thing. But you know, every time the bacterial cell reproduces, it makes a copy of its genome, right? Perfectly normal. But the virus is now integrated into the bacterial genome. So every time this bacterial cell divides, it's going to make a copy of the viral genome as well. So they started off with one cell, divided into, you know, 5 here. And now each one has a copy of the viral genome. So the virus is still replicating. As long as the virus is replicating, it's happy. If the bacterial cell wants to do the work of that replication, it's totally cool with that. But once the bacterial cell starts to slow down in its replication, when it falls on hard times, maybe it's stuck in an environment that doesn't have as many nutrients in it, or maybe it sustains some mutations, some DNA damage, well, then you get induction or excision. And during induction or excision, the cell slows its growth, or it suffers DNA damage, especially DNA damage from UV, and the virus is going to do the process of non-homologous recombination, just in reverse. It's going to jump right back out of the cell's chromosome and leave the cell's chromosome right behind. So now we've had 3 steps of the lysogenic cycle. And now we're going to jump back on to the lytic cycle. But I want to emphasize that this virus can stay in a lysogenic state for as long as it wants. The cell's going to reproduce, and it can reproduce as many times as long as it stays healthy. The virus could stay integrated for an hour, it could stay integrated for a year, it could stay integrated for 100 years. As long as the cell is healthy and reproducing quickly, it's going to stay in there. But eventually, it's going to come back out. This is sort of good for the cell for a while. For a while, the cell is, you know, oh, hey, the virus is here, the virus is being my friend. But eventually, the virus is going to leave it. And the virus is going to leave it in tears, because it's going to jump back on to the lytic cycle. And what are the steps of the lytic cycle that we're missing? Boom! Back onto the lytic cycle with synthesis, assembly, and release.
What about if it makes an endospore?
If it makes an endospore, then the cell goes dormant for as long as it's in the endospore state. The virus can't jump out while it's dormant. So it can't get back on the lytic cycle until after the cell reactivates from its endospore state. However, usually, not always, but usually the sorts of things that cause a cell to become an endospore are things like, lack of nutrients, hostile environment, poisonous or toxic environment, DNA damage. The same sorts of things that are going to cause a cell to go into an endospore are the sorts of things which might cause this, the lysogenic cycle to end and for it to undergo excision or induction. So I can't tell you off the top of my head which is going to happen faster, but it's probably going to be a race. Does the virus jump out and kill the cell, or does the cell make it into the endospore state? Either way, when it gets back out of the endospore state, it would still have the virus in it if it managed to make it in. So that halts the process, but doesn't shut it down. Does that answer your question? Okay. All right, so, yeah, that was video. All right, so I want to switch tacks now and talk about the classification of animal viruses and how animal viruses work. Animal viruses are much more varied than bacteriophages are. Here, I'm going to turn on the lights so that not everyone falls asleep. And so most bacteriophages, and if we call something a phage, we're referring to a bacteria virus, most of them are based on that motif of a sort of moonlander, spidery-looking thing. But you get a lot of different shapes for animal viruses, and you have a lot more of the optional sorts of things. You have more of them using a wider variety of enzymes. You have the presence of envelopes. So most bacteriophages are DNA viruses. Not all of them, but most of them are. Whereas with animal viruses, you get DNA and RNA and about equal, and you get some that are like retroviruses that are just weird. So just to sort of give you an example of all of the different shapes that there are out there. There's a bunch of different shapes. You can have, so this is a little, I can't quite see it perfectly here, but these ones here that are sort of look like, kind of like if you've ever seen the Epcot Center ball, where it's like this big ball made out of a bunch of triangles. So that's called a, so those are called isometric viruses. That's a very common shape. It's a very regular shape. You can also have filamentous or rod-shaped viruses that look like big long rods, often with a little curly Q at the end. They're really long and really thin. You can have bullet-shaped viruses, which are sort of like flat on one side and then they go out and they curve on the other. And you can have what are called pleomorphic viruses, which change shapes or have no defined shape. Those are the common ones. There are some other ones, but those are the really common ones. You also can have an envelope on or an envelope off. So an envelope on virus is called a... Come on, guys. It's in the word. An enveloped virus. A... Naked virus? Good. Naked virus is the one with the envelope off. You can also have spikes, which are an optional feature. So like this is a naked isometric virus that has spikes on. This is a naked isometric virus that doesn't. These spikes are sort of protein appendages, and these are often used for attaching or entering a cell. So these are often going to be how the virus attaches to a cell, how it enters in, especially if it's a naked virus. So, we talked about the two cycles in bacteriophages, the lytic and the lysogenic. Eukaryotic viruses, and animal viruses in particular, are far more complex. And they don't have just two states. Like, it's not quite as binary. They have a bunch of shades of gray. But they do tend to follow either a pathway which is similar to the lytic pathway or a pathway which is similar to the lysogenic pathway. And then there are multiple different ways that they can do each of those things. But if we look at viruses from the perspective of us as humans, the guys who are getting sick, then there's sort of two generalized life modes that viruses, and really all diseases, can take. They can be acute or persistent. An acute infection, an acute disease, is what we usually think of as being kind of the typical disease pathology, right? So this is like the flu. You get the flu, then you get sick, and then you're sick for a little bit of time, and then you either die or you get better. And then once it goes away, it's gone. That's an acute infection. They usually, you get them, you get sick, and then you get better, and then you're done with them. That's sort of analogous to the lytic cycle. The other mode that viruses or other diseases can take is a persistent infection. A persistent infection can take multiple modes, and we'll cover each of them, but basically this is going to be an infection that's with you for a long time. It's probably not going to kill you, at least not quickly, but it's going to be with you. And with you. And with you. And with you. For a long, long time. Probably the rest of your life. It's also important to note that not all viruses, at least in animals, not all viruses actually cause disease. They're all pathogens. But a disease, the disease state, is when you develop symptoms. It means dis-ease. Something has put you ill at ease. And there are some viruses with some people that, you can catch them and pass them to other people, but you never notice that you have them. Like some people who get, what's the disease responsible for mono? That's, okay, let's use HPV. Some people can get HPV, and they never express any symptoms. They can still pass it to other people, but they never actually become sick. Some people get HPV, and they develop genital warts. Some people get HPV, and they develop cervical cancer. But a lot of people, it just doesn't affect. This is called a state of balanced pathogenicity. When you have the disease, but it's not, or when you have the pathogen, but it's not causing an obvious disease. It might be causing a little bit of a drain, like you'll feel a little bit more tired than you would otherwise, but nothing you can really put your finger on. So that's that's kind of off by itself. It's not really causing a disease. If it does cause a disease, the disease can be either acute or persistent. Yes?
What about a disease like chickenpox?
We're going to get to that.
Okay.
That's persistent, by the way, but it's one of my specific examples. So acute infections are short in duration, generally speaking. Here, this is where you get infected. Infected is not when you get sick. It's when the disease passes your barriers and gets into your body. So this guy got infected here, and then the virus replicates until they get sick, and then they start to develop symptoms. And the symptomology, they're sick for a while, and then eventually they either die or their immune system catches up to the disease and starts beating it down. And you have less and less and less and less and less virus until it's gone. And then it's gone. It's not around anymore. This is flus, colds, measles, a lot of diseases. A lot of the classic diseases are in this category. And generally speaking, you're infectious from about, well, you're infectious throughout the whole period. But you're most infectious from like right before you show symptoms to like right before your symptoms go away. But in theory, you can infect somebody anywhere throughout that cycle. So I want to go through the basic steps of the acute infection. These are going to look a little bit familiar because they're very similar to the steps of the lytic cycle. Step 1 is attachment. Attachment is basically the same, right? Virus is floating around, just sort of randomly bumping into cells, and if it sticks to a cell, it can infect it. Like it bumps into this cell, let's say it's a hepatitis virus, it's a liver virus. It bumps into this cell and says, no, you're a skin cell. Bumps into this cell, says, no, muscle cell, not interested. Bumps into this cell and says, ooh, liver cell. Grabs on. Okay, what was step 2 of the lytic cycle?
Entry.
Penetration. Here, it's going to be entry. Slightly different. With the bacterial cell, the capsid could not enter the cell because of the cell wall. So it had to penetrate its way through the cell wall and inject its DNA in. Do you have a cell wall? No. So there's nothing stopping this virus from getting in. So basically these little keys bump up against the locks on the cells, and then we have basically 3 ways it can get inside. If it's an enveloped virus, so the 2 ways enveloped viruses can get inside are shown here. And this one here on the left is, it attaches, and then its envelope, which if you'll recall is made out of membrane lipids, just like the cell membrane is. This envelope fuses with the outer cell membrane, and so it goes, and then the envelope becomes a part of the cell membrane, and the virus passes in. The other possible way is that the entire virus could be endocytosed. So here you see on the right, the virus lands, and then this cell membrane kind of invaginates and takes it in. And here you have 2 membranes surrounding this. This is the virus capsid. This is the viral lipid envelope. And this is cellular membrane material on the outside. And then, once the virus is inside, the cellular envelope, or the viral envelope and the cellular membrane fuse together and sort of peel off, and the virus is released. If it's a naked virus, let me see if I have that. No, that's budding. Okay. If it's a naked virus, then usually what it does is it's very similar to this method, except the virus itself, the capsid itself, is going to dock in. It will be taken in and surrounded by, through endocytosis, it will be taken in and surrounded by a cellular membrane into a vacuole or a vesicle. And usually, most naked viruses are going to have spikes or some sort of protein on their surface that they'll then shoot out, and it will pop the envelope around them. And that's how they'll get free into the cell. But entry is very similar. The difference in entry between bacteriophages and animal viruses is, first off, you have to deal with the envelope if there is one. And secondly, the entire virion, the entire virus particle is taken into the cell. That means that we need a second step, one that wasn't in viral, or in the lytic cycle, uncoding. The virus gets all the way inside, and then it lets its capsid go, and it stops being a virus particle. Then we get replication of nucleic acid and protein. This is basically the same as the synthesis step in the lytic cycle. Maturation, which is essentially the same as assembly, but can sometimes involve some enzymatic activity. Cell lysis. This is cell lysis or budding, I would put down. Cell lysis or budding, which is basically the same as release, but there's a little bit of a difference there. The difference there is that with lysis, again because of the cell wall, in bacteria, the whole bacteria gets blowed up all at once, and the cell is released. Or not the cell, the viruses are released all at once. With mammalian viruses, usually the virus is assembled, and then on its way out, it will sort of bud and pick up new keys. So instead of the entire cell blowing up and releasing a ton of viruses at once, you get a sort of dribble, drabble of like blop of virus, and viruses will sort of leak out over time. This still will eventually kill the cell because that membrane is being taken away faster than the cell can make new membrane. So eventually, instead of happening all at once, it will happen over a period of time, but eventually the membrane will get thin, the cell will get fragile, it'll get depleted of all of its resources, and it still dies. At least with acute infections, they usually do. So release, slightly different because you have this budding mechanism. And of course, since we're multicellular creatures, it doesn't end with the death of a single cell. It has to spread from one cell in your body to all of the other cells in your body. And then it has to spread outside you, and you have to infect someone else. And that's the complete life cycle of an acute infection. Questions on that? All right, persistent infections. There are basically 3 different types of persistent infections. The first is a latent infection. You asked about chickenpox. Chickenpox is a classic example of a latent infection. You get it once, and you're sick for a while, and it goes away. But it never leaves you. It never actually goes away. It hides out in your body and goes into stasis. And then, sometime later, could be a year, could be a decade, could be several decades, but eventually it can pop back out. And when chickenpox pops back out, it reemerges as shingles. And instead of having a full-blown breakout over all of your body, it'll usually emerge along one nerve, because the nerve cells are where it hides out. And you'll get a breakout of hives, of shingles, along all of the skin that's associated with one of your major branch nerves. Other examples of classic latent viruses are herpes, and pretty much all of the herpes family of viruses. Chickenpox is in the herpes family, but yes?
So shingles is really contagious, correct?
It's chickenpox.
Oh, okay.
So it depends. If you have not had chickenpox, you can catch chickenpox from somebody who has a shingles outbreak.
That's what I was going to ask.
That's certainly true. If you have had chickenpox, for some reason, exposure to somebody who has chickenpox or who has shingles seems to precipitate your own outbreak, but it comes from the viruses that are latent in your body. You haven't exactly caught it from them. But it seems to that the reinfection incident does something to kind of wake up or revitalize your latent shingles, and that isn't a process that's very well understood. Do you have a question here?
Yeah, I was just going to ask whether or not the cold is persisting, considering you have to get an annual flu shot.
No, that is due to viral genetic alterations, which it looks like I probably won't have time to talk about, but I'll post. But I want to really quickly get through the other two types of infections. The second type is a chronic infection. This is where you get infected, the virus is produced in your body, and the virus continues to be produced in your body for the rest of your life. Hepatitis B is your classic chronic infection. You get infected with it, your liver cells produce hepatitis B virus. Very shortly thereafter, like you get tested a month after, you've got Hep B virus in your blood. You get tested a year after, you've still got Hep B virus in your blood. You get tested 5 years after, you've still got Hep B virus in your blood, or Hep B virus in your blood. Now, as you get, as you carry this for longer, they do more and more and more damage, so you get worse off, but the level of virus in your blood is the same. Last is slow infections. This is where you are infected initially, and then the amount of virus affecting your body slowly increases over time. So you start off and the virus is barely detectable in your blood. And then it's a little bit more, and then a little bit more, and then a little bit more. And then years later it's more and more and more until there's a whole ton of it. Retroviruses are classic slow infections. HIV is a combination latent and slow. And let me show you. Ooh, okay. Because a lot of people don't actually know this. When you're infected with HIV, you show symptoms almost immediately, within a few days. But they are almost exactly the same as cold and flu symptoms. So you get HIV, you get AIDS basically, and you feel like you've got a cold for a couple of days and then it goes away as your body fights off the initial infection. And then after that, it sort of slowly builds up until it turns into full-blown AIDS. And that's the third type of persistent infection. So you can have latent, it comes and it goes. That's herpes, right? Herpes, you get it, and then it goes away for a while, then you get it again, then it goes away for a while. They say a diamond lasts 1,000 years, but herpes is forever. And it stays with you for the rest of your life and just shows back up every so often to say, hi, I'm still here. You have chronic, which is like hepatitis B. You get it, and it just stays with you at a constant level. And slow, which gradually increase over time. And these are the three main modes of persistent infections. Okay. So, quick question. Was anyone here planning on coming to my office hours today? If no one is planning on coming to my office hours today, then I'm probably not going to hold them because I'm illegally parked right now, and I don't want to get a ticket. But if there was someone coming, I would repark my car and find some way to do that. But if no one's coming, then yeah, then that's fine. Oh, I'm parked in the ASU parking lot. I just don't have a permit.